Hi there, this is Shayan. So we have learned the technique for microscopy. What is microscopy? For microscopy, three types of lens are used that we have learned. And then next thing we have to know the microscopy and the culture surface for which type of experiment we will use which type of microscopy. That is very important. So that's why this video is for microscopy technique and the culture surface. So we have to find the perfect match, which type of culture we need, which type of microscopy. That is the questions. So let's say you are deciding to make an experiment on the cell imaging for a live cell imaging. Which type of microscopy you will use, the inverted or the upright one? So that is a very important question you have to know before the experiment, because normally in maximum case you don't have that type of microscope in your lab. You have to go to central facility. So if you don't decide before and after doing experiment, you have to pay them, and then if you don't get any result, then your PI will be angry with you. So it's very important to know beforehand what type of microscopy you have to use, inverted or upright, for which type of experiments. So if you are searching for this question, this video is for you. Upright microscopy is actually okay. So let me mention one thing. In microscopy, again taking reference from my another video, which is based upon the compound microscopy. So we have came to know that there are three types of lens. One is the objective lens, one is the condenser lens, one is the eyepiece at your eye, near your eye. So this condenser lens has no use to magnify your object or has no use to resolution. Okay, it has the role to resolution that, but in that case, magnifications condenser lens has no use. So condenser lens actually used for converting the lights in a very well-mannered focused light beam, nothing else. But to magnify your object, so light coming from your specimen is going to be magnified by the objective lens, which sits near your specimen, and eyepiece also is used to magnify the image what you can see, and that is near your eye. So two types of lens are actually used to magnify the image of your object. That is, one the objective lens near the specimen, and one the eyepiece that's near you. So this type of lens actually have used. Now this objective lens is very, very important for a microscopy. So this objective lens can be above the plane of the specimen, can be below the plane of the specimen, and based upon that you can say that is upright microscope or inverted microscope, and that has its own advantage and disadvantage also. So for upright microscope, you can see here, let me take the laser. For upright microscope, you can see here, this is the picture of a microscope which is upright in manner. So here you can see the condenser lens will sit here, because the excitations of the specimen is going to be made from the below the specimen plane, and you have the specimen in this position in the specimen, and next you have the objective lens and the eyepiece. So objective lens is actually above the specimen plane. So all the light are coming from the specimen after excitation, or all the image which is going to be formed, that is going to be seen from the top. So light coming from the specimen is going to be seen from the top of the specimen. That's why it says specimen observed from the top. That's here we have shown in focus that this is the condenser lens, and excitation is being used from the bottom surface, and your light coming out from the specimen is going to be collected from the top of the specimen plane. Now what is the advantage and what is the disadvantage? So let me tell you first which type of specimen you will use for upright microscope. For a tissue section or fixed cell imaging, in a single word, for fixed cell imaging and tissue section imaging, actually upright microscope is highly recommended. So the point is that with tissue section and fixed cell, what happens and what people do for sample preparations, they take a slide and put the cell solutions and then put a cover slip. So you pack you pack means you close all the cell solutions inside the cover slip and the slides. So your cell solution is squeezed, cell solution or sample is squeezed between the slide and the cover slip. Now when you are culturing the adherent cell, okay, so sample is squeezed between them. Now you want to see the specimen from the top of the specimen, then you can use the upright microscope. But in the case of live cell imaging, where actually you have to change the media for a long time imaging, because live cell imaging goes on, let's say 1 day, 2 days, so we have to change the old media and have to put the new media. So in that system is actually not accessible in this upright microscope. That's why live cell imaging you can do, but fixed cell imaging or tissue section imaging, where your sample is squeezed between a slide and a cover slip, that kind of system can be imaged by the upright microscope. And another main advantage is there for the upright microscope is that another advantage of the upright microscope is that you can dip your objective in your specimen. So here actually what happens, if you have a tissue section, you want to go as much near as the tissue surface, then you can go with the upright microscope, because your objective, you can dip your objective into the specimen. Okay, and also for the fixed cell you can dip your objective in very close to the specimen. So they first in the place where specimen is kept, they do what? They first keep the specimen, means squeezed specimen between the slide and a cover slip, and then they put some water or oil. If you have oil immersion objective, then you put oil on the cover slip, and if you have water immersion, then you put water, and then you image it, means taking light from the specimen. So that's the point. So advantage is that you can dip your objective in your specimen, and that is useful for the tissue section imaging and fixed cell imaging, but not for live cell imaging. So but live cell imaging is very important. Live cell imaging is very important to, let's say, investigate the entry of a cellular entry, investigate the cellular entry over time of a nanoparticle of a biomaterial inside a cell. So that case live cell imaging is very important. So for live cell imaging, there are another microscope called inverted microscope. Okay, so the arrangement inverted microscope is a special in this case is that you can change here the media if you want to do for a long time imaging. So here what happens, you can understand from the name inverted. Inverted means objective lens in upright microscope, objective lens was top of the specimen plane, and that will be opposite in inverted microscope. Your objective lens is below the stage and pointing up, so all the light coming out from the specimen is going to be collected from the below of the specimen. That's how shown here. That's how we have shown here. This is the 100x objective lens, 100x objective lens, and these are going to collect all the light coming from the specimen and which are below from the specimen plane. So what happens, this is useful for the cell culture, for the live cell imaging. Why? Because let's say you are doing cell culture for an adherent cell. So you will culture it in a cell culture flask. Now when you are culturing, all the cells actually will sink at the bottom of the cell culture flask. So all the adherent cell, and it's as we are considering the adherent cell and like a muscle cell, etc., then it will adhere at the bottom surface of the cell culture flask. And as we are not considering the non-adherent cell like RBC, so that is out of the discussion now. So as we are considering the adherent cell like muscle cell, it will adhere in the bottom surface of the cell culture flask. So all the cells or whatever particles, fluorescence particle you have inside the cell, all the things you have at the bottom plane of the cell culture flask. So if you can image the bottom plane and take the light coming out from the plane in below the specimen plane, then you can get a very clear idea what is going on inside the cell. That's why objective lens is placed here in inverted microscope, not in the top but in the below, because you want to take all the light coming out from the specimen from below the plane of the specimen. And this is useful because all the cells are adhered in below plane, in the bottom plane of the cell culture flux. And another special advantage is there, as the cell culture flux you have the below plane, bottom plane, where the cell are adhered, and upon that you have the cell culture media. Now for a long time cell imaging, let's say 1 day, 2 day, 3 day, people do like that, and then you can change the old media and put the new media, and that is quite meaningful for this system, and that accessible also for this system. That's why this is highly recommended for the long time live cell imaging. Inverted microscope is actually highly recommended for the long time live cell imaging, and cells are observed through the bottom of the cell culture vessel. That's the real advantage of the inverted microscope over the upright microscope. Now upright microscope is actually not good. There are serious disadvantage of the upright microscope over the inverted. What happens? Upright microscope, you have a longer working distance. Okay, you have a longer working distance, so that actually leads to lower resolutions or weaker fluorescence signal. When you are taking light coming out from the specimen at a distance, at an appreciable distance by the objective lens, then obviously some signal coming out from the specimen is going out of your objective. So objective is not going to be able to capture all the lights coming out from the specimen. So that is a real advantage. You can get weak fluorescence signal, and also resolution matters here, because here the distance decides the resolutions. So we will see how distance decide the resolutions from another video. You can follow that one. So that's the real disadvantage of upright microscope. That's why people prefer the inverted microscope over upright for many cases. Hope you understood the video, hope you understood the concept, and thank you for watching. If you have any questions, please feel free to comment in the comment box or ask me in the following mail ID I have given in the description of the video. Thank you.